Clustered, regularly interspersed short palindromic repeats is a very long acronym that is abbreviated to CRISPR, which is certainly a CRISPR way of talking about the adaptive immune system found in about 40% of all bacteria and 90% of archaebacteria. And it was discovered from the sequencing of lots of prokaryotic genomes, and you kept seeing these repetitive regions. So in black here are all of these different repeats, and in front of the repeats were all these open reading frames. And so groups got into studying what these CRISPR loci were. What were these clustered repeats all about? And what it is a system with which to detect bacteriophage genomes, detect and cut them, and also save fragments of those phage genomes into these repetitive arrays as a record of previous infection, and so you can target them again. So there's a bit of a chicken and egg situation goes on here, but if the CRISPR-Cas system has identified a viral genome and cleaved it, and therefore inactivated that phage infection, and cut it up into small pieces, some of those small pieces, which are called spacers, get inserted into this CRISPR array. So all these different colours here are fragments of phage genomes that this particular bacterium has cleaved before in its evolution. This repeat array, this CRISPR array, is then transcribed as one long pre-CRISPR transcript, a polymerase II transcript, which has got all these repeats in it. Those repeats, each one of those repeats binds this other tracer RNA to form a double-stranded DNA sequence here, and then RNase 3 then binds to this duplex of RNAs and cleaves them. So each one of these spacer and repeat regions, called a CRISPR RNA, gets separated from this pre-CRISPR RNA transcript. So you end up with lots and lots of CRISPR RNA, tracer RNA fragments. These are then bound by Cas9, and Cas9 is an RNA-guided DNA endonuclease. So these double-stranded RNAs bind to Cas9 and program it. They activate it for searching the genome. Cas9 searches the genome for short DNA sequences called protospacer adjacent motifs, or PAM sequences. So each one of these spacers that we talked about before, these phage sequences, these spacers, when present in the CRISPR RNA, is now called a protospacer. And Cas9 needs to find a sequence in the genome called the PAM, which centers it, it allows it to sit onto DNA. Cas9 then unwinds the strands, and if there's a match between the protospacer it has in its loaded CRISPR RNA, if that matches the genomic target, then the unwinding continues on, and Cas9 will then cleave that target. So we can look at this in more detail. Again, we're looking at the natural CRISPR system, and this is a class 2 CRISPR system. So we have Cas9, and it loads up this duplex of RNAs of the CRISPR RNA, which contains the protospacer, which is 20 bases, which is supposed to match a phage genome. And then that's paired up with the tracer RNA. Cas9 then searches the genome for short PAM sequences. These are often just 3 or so bases long, and it then unwinds the strands at these PAM sequences and starts to see whether there's a match between the CRISPR RNA and the genomic target. If there is, this unwinding continues, this bubble gets bigger, and when it reaches its full length, and you've got this long R loop formed, so an R loop describes this RNA DNA duplex here, and Cas9's holding the other DNA strand separately. When it's got far enough, then the cleavage domains of Cas9 are activated, and the Cas9 systems used in most laboratories create a double strand break, which is blunt ended, and it cuts 3 bases away from that PAM sequence. So very predictable cleavage. Thanks for watching. Let us know in the comments below what you thought of it. Please give us a like, and certainly think of subscribing. And we've got a lot more content on this channel, which you can see in the playlists coming up. Thanks.